That was a fateful day in the life of the last Russian emperor, Nicholas II, and his family, who had been held captive in a nondescript house in Ekaterinburg in Kiev province. Years after their execution, the pit where their bodies had been dumped was located and their skeletal remains exhumed. Though Russian and American forensic anthropologists confirmed that the skeletal remains were those of the Russian imperial family, doubts persisted. The doubts were set to rest when two molecular biologists, Peter Gill of Britain and Pavel Ivanov of Russia, provided the DNA evidence. They found that the DNA extracted from the cells of imperial bones matched with one another and with that of the living relatives of the Russian imperial family. That was one of the examples of the application of DNA fingerprinting, a technique developed in 1984 by Sir Alec Jeffreys, a British geneticist. This technique relies on the DNA, that is, deoxyribonucleic acid, which is responsible for transmission of genetic information from one generation to the next.
The DNA is present in thread-like structures called chromosomes. In human cells, excepting sex cells, there are 46 chromosomes, 23 inherited from father and 23 from the mother. Every chromosome contains proteins and one extremely long DNA molecule.
As described by James Watson and Francis Crick in 1953, each DNA molecule contains two strands entwined to resemble a double helix or a spiral ladder. Each strand of the ladder has a long chain of nucleotides, and each nucleotide is made up of 3 subunits. A 5-carbon sugar molecule called deoxyribose is one of the subunits. Attached to one of its ends is the subunit of a phosphate molecule. These two make the backbone of the helix. Attached to the opposite end of the deoxyribose is the 3rd subunit of a nitrogenous base. It is either A, that is adenine, or T, that is thymine, or G, that is guanine, or C, that is cytosine.
It is at these bases that nucleotides of one strand are joined by weak hydrogen bonds with the nucleotides of the opposite strand of the helix. The electric charges and structure of the bases allow them to pair in a restricted manner.
Due to their structure and nature of electric charges, only adenine bonds with thymine. Similarly, cytosine bonds with guanine, forming millions of base pair sequences.
The specific length of a sequence that codes for a stretch of amino acids is known as a gene. Only about 3% of the total sequences in the genome functions as genes. The rest of the sequences constitute a repetitive class of DNA with no known function. Most of the base pair sequences that constitute functional genes differ very little from one person to another, except in regions called tandem repeats. In these non-functional segments, the base pairs are repeated from a few to a few hundred times. Depending on their length, the tandem repeats are called mini-satellite, microsatellite, and single nucleotide polymorphisms. It is in the nature and number of repeats that the DNA of one individual differs from the other.
For instance, in between a set of functional genes of one individual, there could be a GTG base pair sequence repeated 5 times. In another individual, this sequence could be found repeated 7 times or any number of times. The presence of such tandem repeats have helped molecular biologists develop DNA fingerprints which resemble barcodes. DNA fingerprints are unique. Hence, these are being increasingly used in solving cases of child swapping, homicide, and extramarital relations.
Suspicion of extramarital relations often leads to disputes over offspring's paternity. In such cases, the inheritance of equal number of chromosomes of both the parents provides the basis for DNA fingerprinting. Therefore, DNA is analyzed to examine whether the child has copies of the DNA of both the mother and the father.
This little one's family, for instance, is in the grip of such a dispute. Unwilling to accept the little one as his child, the doubting father approaches his counsel for legal advice. The matter is finally referred by the family court to molecular biologists for DNA fingerprinting.
We have one more case for DNA fingerprinting test. This is a disputed paternity case.
Let us see how molecular biologists develop DNA fingerprints and what do the prints suggest. The first step in DNA fingerprinting is the isolation of DNA from body tissues such as blood. In child swapping and paternity cases, about 5 to 10 milliliters of blood of the putative parents and the child is collected for analysis. In blood, the DNA is located only in the nucleus of the white corpuscles. Therefore, the red blood corpuscles which lack nucleus are lysed by adding hypotonic ammonium chloride solution. For better lysing of the red blood corpuscles, the sample is kept in ice and then centrifuged to pellet the white blood corpuscles needed for isolation of DNA. From the centrifuged sample, the red blood corpuscles are discarded. The white blood cells are then lysed with a buffer, a detergent, and an enzyme called proteinase K. This enzyme digests the proteins, freeing the DNA. To maximize this activity, the sample with these chemicals is incubated at 56 degrees centigrade for about 1 hour. After incubation, a high concentration of sodium chloride and chloroform is added and again centrifuged. This precipitates proteins in the lower chloroform layer. The upper aqueous layer, which contains the DNA, is pipetted out. To this layer, alcohol is added to precipitate DNA. The DNA now shows itself as a white filamentous clump. The DNA clumps are carefully pipetted out. These are then washed, dissolved in a buffer, and incubated overnight at 56 degrees centigrade to obtain pure DNA. Now begins the second stage of checking the purity of DNA extracted from the blood samples. For this purpose, molecular biologists load the DNA samples in cuvettes. These then are placed in ultraviolet spectrophotometers, which help check DNA purity.
From the pure DNA so isolated, fingerprints could be obtained through either the VNTR, that is variable number tandem repeat approach, or the STR, that is short tandem repeat approach. If it is the STR approach that is chosen, the third stage in DNA fingerprinting would be the amplification of DNA through polymerase chain reaction to obtain millions of copies of the specific short target sequence.
For amplification, a primer, DNA polymerase, and free nucleotides are added to about 250 nanograms of pure and diluted DNA sample. The microfluids are placed in a polymerase chain reaction machine programmed for repeated heating and cooling cycles. During the heating cycles, the two strands of the DNA separate. During cooling, the DNA polymerase makes a copy of each of the strands, producing two identical molecules. "30 such cycles produce about 30 billion copies of the target sequence. Electrophoresis of the amplified DNA fragments is the next step. Electrophoresis is a means of separating biological molecules by filtering them through a gel with the help of an electric current. The gel, a mixture of acrylamide and bisacrylamide, is poured between two glass plates. On the top end of the gel, welds are made using a comb. The plates with the gel in between are mounted on the electrophoresis apparatus. The amplified DNA of the putative father, mother, and the child are loaded into the wells. When a high voltage is applied, the DNA, which is negatively charged, moves downwards through the gel, smaller fragments moving faster than the large fragments. Thus, the DNA fragments in each track clusters at different positions along the gel, producing a distinct pattern of bands. It is these bands that are referred to as DNA fingerprints. With that begins the last stage of DNA fingerprinting, the visualization and interpretation of prints. Molecular biologists compared the bands of the father, mother, and the child whose parentage is in dispute
Now, all are
to find out
heterozygous for this
whether the
loci. As
child
you can
has
see,
the
two
same
bands.
genetic prints
Had they
as
been
the
homozygous,
father
there would be
and
only
mother.
one band. Now, the child shares this allele with mother and father because it's a common allele present in both father and mother. Based on this loci, we cannot exclude the father from the disputed paternity. Now we go to the second loci. Now here
With the
also,
DNA
one
evidence,
allele is
molecular
common.
biologists have thus helped settle the dispute, reuniting the family happily.